So, in the last class we have discussed about the high resolution electron microscopy principles, and I said that we will discuss about the image simulation today. As we know that high resolution electron microscopy gives you images of the atoms or the columns of atoms to the resolution level of Armstrong or sub-Armstrong. Even it is possible to obtain resolution level of 0.8, 0.7 Armstrong in the microscopes available today. The problem is that interpretation of such high resolution images are really a difficult or daunting task, because image contrast can vary drastically depending on the focus. As we know, I have shown you that defocus is the most important factor in obtaining the high resolution images in the time electron microscope. So, therefore, during the subsequent process, after we receive the images from the microscope, we need to simulate it using the prescribed models available for the crystal structure of the material. If it is a crystalline, if it is a non-crystalline material, then we must have a prescribed model available for the structure of the non-crystalline material like amorphous or any other structure. So, during as you know, during imaging process, the electrons undergoes 3 distinct interactions, if we have to list down in a very short form, and each of these interactions can be simulated in a computer. So, first thing, or first interaction electron undergoes is called the, basically because of this dynamical scattering of the electrons in this sample or the specimen, and this basically this can be simulated. This dynamical scattering is basically because electrons falls on a sample and then can get scattered in different directions, and this is basically it is a dynamical process. So, one needs to apply the dynamical diffraction theory to obtain the exit wave functions of the electrons. Now, this can be done using a technique called multi-slice method. In this method, actually the specimen is sliced into many slices. Actually, specimen is basically taken into many different small slices, and then this slices are basically taken at a normal to the electron beam or incident beam, and then this, after we obtain these slices, we can simulate the electron interaction, dynamical interaction of the electron with the material by using different methods. There are methods like reciprocal space formalism, or fast Fourier transformations, or real space approach, or block wave approach, depending on how precisely you want to determine the interactions. Now, I do not have time to talk into each of these techniques very precisely because of the time constraint, but nowadays any conventional textbook on the transmission electron microscopy will provide you all the knowledge base available for these techniques. The most important technique which you day to day apply in the high resolution electron microscopy is called fast Fourier transformations. In this technique, we basically use the actual high resolution image which showing the columns of the atoms as a real space image, and then Fourier transform this real space information and obtain the diffraction pattern. Then compare the diffraction patterns with the ones which can be derived from the structural model available, provided we know this structure of the material very precisely, and then by comparing it we can get many distinct information regarding the interaction of the electrons with the material. An input to such a multi-slice method obviously has to contain different parameters of the specimen or the object, like unit cell, the position of the atoms inside the unit cell, the what is called thermal factor, Debye-Waller factor, because of the vibration of the atoms as the temperature increases, and the orientation of the specimen or orientation of the crystals or the object, as well as the thickness. So, this itself is basically not an easy simulation. As you understand, there are so many parameters which goes into the simulations, and if we do not have distinct information or distinct values of these parameters like thickness of the specimen the object orientations, the values of the Dybala factors very precisely for the material which you are studying, we do not get very good simulated picture. Now, results of this calculation actually gives us the wave function. As we know, wave function is this important result of this. Wave function at the exit plane of the sample is obtained by using the multi-slice method, and in the second step, once you obtain these wave functions, the formation of the images in the electron microscopic the time electron microscope can be simulated using different equations which I have discussed. These equations actually are based on the transfer functions. So, transfer functions actually are to be known a priori for a particular set of microscopes. Once you know the transfer functions and related equation, how this the points in the specimen is getting transferred to the image plane, then we can basically obtain the instrumental factors in the image. So, therefore, in this simulation, second step simulation, one is to provide all the information regarding the instrument or the microscope which you are using for obtaining highly spectromicroscopic images, and this includes obviously the spherical aberration constant, the defocus, as well as even if it is possible to give the information regarding the currents of the different lenses subsequent to the objective lens. Finally, in the third step, electrons intensity of the electron beams which are coming at the image plane because of this interaction can be calculated by squaring the wave function, and this can be displayed like a monotone or half tone image, basically half tone image on a high resolution screen on a computer. So, these three steps are normally done nowadays in all kinds of computers available, where the commercial softwares actually allows you to run these different steps and provide you actually the image which is can be compared with the actual image. Now, in practice this is actually routinely done in the even while doing the microscopy, one can run these things, one can use these functions which are available in the software in the computer and compare and get information. Or otherwise, if the structure looks to be very complicated, then one needs to go back to the structure, modify it, and then obtain images which can be then compared. But these simulations obviously are dependent on many input parameters, as we have seen. The most important input parameters are the specimen and the microscope itself. Now, as far as the microscope is concerned, one can actually give these input parameters very precisely, the values of the CS or the specular aberration constant, or even the defocus values can be provided. But as far as the specimen is concerned, it is very difficult to provide the actual values of the input parameters, like orientation of the sample, the thickness of the sample, where the image is taken, and many what's called other parameters. So, that is why many people actually what they do, they initially obtained this information or measure the thickness of the sample very precisely using convergent electron diffraction technique or some other technique available, and also determine the orientation of the sample very precisely and use this as input, precise values of this are used as input to the simulation. So, that one can obtain what's called good images. For reasons these are sometimes available, for reasons these are sometimes not available to do the simulations. If you are lucky enough, you are having a good microscopic team, then you can get this information very easily from your own experiment or from your colleague experiments during the whole set of time electron microscopy, and then you can run these simulations. At the same time, for the machine, as I said, it is possible to provide the precise information of specular aberration constant or defocus values, but many times we do not rely on that, the defocus values which are provided. Rather, what we do is that we take a series of defocus images from the high-resolution electron microscope and then compare it with the simulated images. So, therefore, in a nutshell I could basically show you a simple simulated picture in the slide, where this is the specimen. You see there electron from the gun falls on the specimen after going through different lenses, and then interaction basically dynamical in nature, and then is basically passes through the other lenses in the microscope like intermediate projector lenses, and finally we obtain an image in a CCD camera nowadays. And to make life simpler, we always get a defocus series of images, where defocus is very of the optical lens is very what's called systematically, and images are obtained as black and white dots, as you have seen in some of the pictures, and these images are fed into the computer. Once this image fed in the computer has the software inbuilt, and these softwares are basically runs based on these three principles which I have just now told you, and then reconstruct the original image by comparing with the model available, and finally it gives you a potential map. So, that is actually the cycle in which we simulate the images and obtained finally a most comparable image. But you understand from this whole process, because there are a lot of input variables which can be changed, or the few variables, and also the complexity of the whole technique tells you that it is not an easy one to basically get information out of high-resolution images. That is why nowadays people provide these high-resolution images in the different papers or the books, just for the sake of increasing the quality of the images. Many times we find that the exact information from these images are lacking in both in the papers and sometimes in the books also, because of this problem of simulating these images and getting the quantitative information. So, obviously qualitatively one can explain the images using such software, but obtaining quantitative information requires another kind of simulations which are much more complex and probably out of the scope of this particular course. So, I will not go in detail of this, but just to give you an idea that how complex is this simulations, I am just telling you this aspect, that be careful while comparing the actual images with the simulated ones. Sometimes this may be misleading. So, therefore, you need to first learn how to obtain good image in the electron microscope, like good ones in such as Titan, and then compare these images with the best possible software available in this world to extract quantitative information about the micrographs. So, with this I close the high-resolution electron microscopy portion of this course, and I move on to the next portion of this course, that is called STEM, Scanning Transmission Electron Microscopy. As you know, scanning transmission electron microscopy has become an integral part of the microscope nowadays. All the modern day microscopes has scanning electron microscopy facility, scanning transmission electron microscopy facility. This has originated from work by Crowley, Crew, and many others, and those who have tried very hard to use scanning transmission electron microscopy. The reason scanning electron microscopy is important is because of these aspects. We can get informations for bright field, angular dark field images, high angle dark field, high ended angular dark field images, which can give us jet contrast. We can also we integrate this STEM with EELS energy filter, electron energy loss spectroscopy, which can be used to obtain energy filter images. One can also integrate this with converging electron beam or nano electron beam diffractions, and one can actually use the STEM to get holographic images. So, I will not be able to discuss all of them in this course, but I will try to give an idea how STEM works and how STEM can be used to obtain different sets of informations. As I said, the concept of scanning transmission electron microscopy is not a new one. it has been employed by Crewe long back, who introduced this whole concept into the electron microscopic community, and he actually used first time showed that for scanning time electron microscopy you need to use most notably the field emission guns. So, in a scanning times electron microscopic images, what we basically do is like this. We have basically a fake source or FEG source, what we can say, which has a very high brightness and a very small Lange spread. This FEG source can be focused by using the objective lens onto the specimen. Obviously, the FEG source, when it is focused onto the specimen by using this kind of objective lenses, the beam will be demagnified, electron beam, and basically this beam is called convergent beam, and once the convergent beam falls on the sample or the specimen, it diffracts and gives you diffraction disk, and this diffraction disk can be either transmitted electron beam or diffracted beams. Now, one can actually use different detectors below this specimen to obtain the images using this that is called either transmitted beam or the diffracted beam. So, that is basically idea. So, it is shown very nicely here in the second picture, where you can see this is a source or electron beam, which is then it is can be scanned over the sample by using a deflector, which is there in normal scanning electron microscope, and this image is falling, this beam is falling on the objective lens, which focuses or demagnified the electron beam to a very small size beam called convergent electron beam on the specimen, and then specimen diffracts, and we can get different kinds of informations which are listed there. So, this is basically the idea. Now, as you understand that as the convergent beam falls on a specimen, some part of this convergent beam will be diffracted and some part will be transmitted. So, if one collect this diffracted beam intensity using a detector, one can form dark field images, or if one collects this transmitted beam using a detector, one can actually display it as a bright field images. So, and then once this beam scans over the sample, and this whole imaging can goes on as the speed of the raster, just like a scanning electron microscope. In scanning electron microscope, the raster scans the electron beam and signals like backscatter electron or the secondary electrons or whatever other signals are generated. These signals are then used to display the image on the computer screen. Same thing can be done here also. As the beam scan on the sample, the diffracted or the transmitted beam can be either of them can be taken by the detector, and we can be then plotted on the computer to obtain an image. So, the first thing you understand, the intensities of the diffracted beams or the transmitted beam which is passing to the sample is totally dependent on the initial intensity of the convergent beam which is falling on the specimen. So, that is why in this kind of STEM configuration, one uses something known as FEG or what is called as field emission guns. Reason is very simple. In field emission guns, intensities of the electron beams are very high, almost 3 to 4 order of magnitude higher than the normal lab 6 the filaments or the tungsten hairpin filaments. So, these beams which are high energy or high intensity beams rather can be demagnified to about nowadays 1 nanometer or even less. In many cases one can go down to 0.5 nanometers also demagnified, and to get a very fine probe of very good intensity of falling on the sample. So, that the diffracted beam which is coming or the beam which are coming out from the exit surface of the specimen can have sufficient intensity to get informations regarding the sample. So, that is why FEG sources are normally used. As I to give an idea, if I take a FEG field emission gun source, 1 nanometer beam has a current of 0.5 nanoamps. So, that is why with FEG bright and dark field STEM images can be easily recorded within few seconds or even at TV scan rate. That is what we want. We want the beam to scan on the sample and then image to be displayed at the same scan rate. So, to do that you need a TV scan rate, and to get a TV scan rate you need to have a sufficient intensity of the beam. So, as I have discussed with you this slide, this looks like very simple, but there are a lot of attachments involved which I have removed for the sake of simplicity of the images. But essentially the components in a scanning transmission electron microscopy is more or less same as a conventional transmission electron microscopic instrument, and not much difference is there. So, except that there is a scanning coil which can make the beam scan on the sample like in raster mode, just like a scanning electron microscope. But there are a lot of advantages using this scanning transmission electron microscopy. So, the real advantage is that the dark field images can be obtained with a very high collection efficiency in STEM as compared to the normal dark field images in conventional transmission electron microscope. This is mainly because that all these scattered electrons outside the incident beam spot can be collected in a STEM mode, and once we collect all the scattered electrons or diffracted beams, the intensity of the diffracted beams is sufficiently high, so that it can be it can give us a very good collection efficiency. That is one of the biggest advantage, and that is why almost half of the imaging detectors in the STEM mode are dark field, like annular dark field, high angle annular dark field. These are actually routinely used to obtain all kinds of different information, which we are going to discuss one by one. Not only that, that is the biggest advantage. Not only that, another important advantage is that in the STEM, in a conventional electron microscope, the conventional TM, normally a 2-dimensional detector such as photographic plates or a CCD camera is used to record the intensities at all image points which are in parallel, like as the beams goes parallel and falls on the screen, the images are recorded. But in STEM, image information is produced in a serial form, like a time dependent voltage or current variation. For many years, in fact, this gives STEM this unique possibility of online image processing to manipulate the image contrast for special uses or purposes. And now we have the CCD cameras available of high quality, high resolutions, and so therefore, this gives us the serial readout or online image processing processes in conventional TM also. However, STEM for the STEM there are many other possibilities exist. Thus, in STEM detectors can be, or in STEM actually several detector can be used simultaneously to produce images from the different signals coming out from the sample. So, there are a variety of these STEM detectors available, and which I can show you here, or I have listed down there. Variety of STEM detectors available to obtain different signals other than this bright field and dark field. So, as this beam falls on a specimen, it's one part of the beam is getting diffracted. That is what is the big this diffracted disc is shown here. Other part is called zero beam, which is not undergoing, supposed to be undergoing, not undergoing any diffraction as forward scatter beam. So, we can put a bright field detector there, and we can put a dark field detector there and obtain conventional bright field dark field images just like that. As the collection efficiency high for dark fields, image quality is better. Even many cases resolution is also better, because the beam which is basically coming out after diffractions, they contain the information which are having very high resolutions. Not only that, one can actually use integrate, as I said, the STEM with the electron energy loss spectrometer, which can allow not only allow you microanalysis of the specimen, a very small area to detect elemental presence or the state of different elements present in the sample or specimen in a small area, but also it allows you to from the images with electron that have lost particular amount of energy. we have to understand that yields basically operates on the energy loss of the electrons. So, as the electrons comes out of the sample exit phase, they carry loss information, because they have electrons have undergone different kinds of amount of energy loss, and this is nothing but because of the inelastic scattering of the electrons in the specimen. And so, therefore, we can trap this information by trapping those electrons which have undergone loss of energy because of inelastic scattering, and this loss can be characterized depending on the type of element present in the sample. So, one can actually use a certain kind of technique in which a specific energy levels can be used to obtain image, and these are all called energy filter imaging. And so, therefore, the what is called as the images which corresponding to electrons that have lost a particular amount of energy can be obtained. And to give you an idea, this is the illustration which I have shown in the very first lecture for the nanocrystalline copper pulse electrodeposited using thiourea. So, it is deemed or thought that thiourea act as a gain refiner by pinning the gain boundaries of the copper, but there is no work or no experimental evidence available showing that thiourea molecule is exactly sitting at the periphery of the copper grains during electrodeposition of the copper. So, to show that, one can actually use what is known as energy filter imaging using yields. As I said, that yields can be used to filter out or to image or to obtain images for the electrons which have lost a particular amount of energy. This is the left side of the picture is basically showing you the normal bright field image, and right side of the picture is taken using sulfur energy edge, which is, that means the electrons which have lost energy corresponding to sulfur energy edge in the yields, and that can be used to obtain the bright field or image. And there you can see this bright lines at the gain boundaries many places. This bright line signifies the presence of sulfur, and as I told you even in the very first lecture, the thiourea is basically a molecule which contains sulfur other than nitrogen, hydrogen and carbon, and sulfur is the distinct part of the molecule. So, the presence of sulfur means presence of the thiourea. So, by doing this kind of exercise, one can actually obtain images to the resolution of the transmission electron microscope, where we can show that where a particular element is present. So, therefore, these are now does routinely. In fact, in catalyst, which is a very important field of research nowadays, one can actually use this energy filter images to clearly signify where a particular element is present or not. So, other than that, in the STEM mode, this is one of the yields is one of the most important attachment to this scanning transmission electron microscope, which many microscope has. The microscope which I showed you, we in our campus do not have these yields, but I have shown you how the yields can be attached at the bottom of the microscope just below the camera. Yields can be attached, and the signal can be processed using a computer. Also, images can be formed in the STEM mode by using low energy secondary electrons or OG electrons or characteristic X-rays rather. So, serial nature of this image signals provides basically possibility of the quantifications basically, and then the information can be correlated with the specimen compositions, crystallography, and as well as the morphology. So, another important detector is basically added into the microscope or into the STEM is known as HARDIF, high angle annular detector. This was basically done or developed by Crewe et al, and they have seen that if we use an annular detector means just like this, Suppose this is the specimen, here as you can see, and this is my convergent beam coming from this objective lens stem, and then obviously electrons are undergoing diffractions. So, if you use a detector which is annular type, so what actually happens is that we can collect the diffraction information in a disk, okay, surrounding by this central circle, and this is what is called annular detector. In fact, there are reasons for it which I will tell you within few minutes time. So, if we can put these detectors at a very high angle means not very high angle. In electron diffraction, you have to remember the scattering happens at a small angle, maybe 1 microradian or few actually microradians or few 1 or 2 microradians like that. So, the high angle means of that nature, not that several couple of 20, 30, 40 radians or so, no. So, so therefore, these detectors can be placed just like this one source here at a little distance from the transmitted electron beams or the 0 beam, and they contain information regarding the atomic number contrast. It has been shown by Cue et al. That basically that intensity of the image which forms by these detectors is proportional to z to the power 3 by 2, where z is basically the atomic number of the element present in the specimen. So, therefore, these detectors, if we collect the information from the detector and display, will tell us chemical compositions of the phases present in the microscope. This is very clear, and this is routinely done nowadays. In fact, the microscope which I showed you has a Hardy detector, and Hardy detector takes this signal which are coming out or coming from the specimen at a little larger angle than the commonly diffracted beam in an annular detector and displays on the computer scheme. So, to show you one such image here, this one I have shown you in the very first lecture of this course. This is basically a picture that one and the taken in a Hardy detector in a microscope where there is a fake gun. So, as you see, this is there are large number of nanoparticles present in this specimen, and these nanoparticles are embedded in a matrix. It is basically here is aluminum matrix, and these nanoparticles are laid in eutectic alloys, and as you can clearly see that in a high angle annular dark field TM image, we can clearly see these particles showing distinctly two phase contrast. One is lead solution, or is tin solutions. Aluminum is shown as a dark. So, as we know that aluminum having very low atomic number, so therefore, jet contrast because of the aluminum will be very low. On the other hand, lead and tin having very high atomic number, so jet contrast will be very high, but there will be distinctly different jet contrast variation in from the tin and the lead, which is seen from the brightness of this different portion of the particles. As I said that the bulk of the particle, as you see on the right hand side picture, is tin, or the cap is basically lead, and lead having higher atomic number than the tin will be looking brighter in the Hardy images. So, this is again taken from our own work. So, one can actually use this particular technique to decipher this kind of informations, and rather to decipher different kind of phases presence. This is exactly equivalent to the backscatter electron imaging in scanning electron microscope, where the backscatter electrons carry the information regarding the jet contrast, and we routinely do in the scanning electron microscope to obtain the jet contrast images, so that we can see different phases very clearly. Exactly same thing can be done in STEM, but at a resolution of high resolution electron microscope. Now, one of the biggest fallout of this particular Hardy detector is this: if we can use this Hardy detector to obtain high resolution electron micrographs. Okay. So, how to do it, I will just explain you within few minutes time. Is like this: suppose you have a very thin portion of the sample or specimen, and you are using the STEM mode, so that the convergent beam is focused onto the particular column of atoms, and obviously as I said you, using good fake guns, one can actually convert the electron beams to 1 nanometer or 0.5 nanometer level. So, if the small beam of very high intensity falls on a column of atoms, and then this column of atoms contains suppose different atoms of different atomic numbers, some of them are like aluminum, which is very low, some of them let us suppose contains holmium or maybe some other very heavy lead, very high atomic numbers. So, obviously, the atoms which are heavy, they will diffract strongly than the atoms which are having low atomic numbers. So, if we take or if we collect this information in hard detectors in a high resolution mode, we can clearly depict that which atom is what actually, whether it is aluminum atom or it is a holmium atom or it is basically a lead atom, one can clearly show on the microscope at the resolution of the high resolution micrograph. So, this is one of the another important thing which normally people do, and if we collect this high resolution images in a hard if mode or in this in the from the hard if detectors serially, then one can build the whole structure of the crystal slowly one by one. Well, nowadays using titan we do not need to do it, because in a titan basically one can get the distinct contrast from different atoms, like one can get contrast from I have shown you the example of strontium titanate in the last lecture, from strontium, titanium and oxygens differently. So, you do not need actually hard detector that way. So, this is another advantage of the STEM. The STEM actually has changed the whole color of the electron microscopy so much. Now, as I said that the one of the what's called advantage of this high angle annular detector is to obtain Z contrast images, but it has its own limitation also. Limitation in the sense that it has to be properly, sample has to be properly oriented, and in many cases we found that very the signals are dependent on the sample thickness. The sample thickness is high, the hard if signals are very good, the sample thickness are small, hard if signals are bad. So, that is why one needs to use a very high intensity beam at the as an incident beam on the sample, that is to be focused by the objective lens very properly on the sample, and so that the intensities of the hard if detector or the hard detector for the thin regions of the sample can be comparable. Another important technique which is attached to the STEM is the convergent beam electron diffraction and the nano beam electron diffraction. Specifically, the nano beam electron diffraction is nowadays widely used. We know that many techniques allows us to form grains or the particles which are very small, suppose less than 5 nanometers. So, in those cases it is very difficult to obtain diffraction information using conventional diffraction like the selective area diffraction pattern. So, one needs to use the CBADE or the convergent electron diffraction or specifically nano beam electron diffraction. To give you an example, suppose there is a particle here small which I marked there in the image, size is approximately 10 nanometers. So, to obtain diffraction information from such a small particle, one needs to use a very fine probe, and that kind of fine probe can be obtained in a STEM mode. So, that the beam which is highly convergent, size of the beam of the order of suppose few nanometers can be allowed to fall on this particle, and then diffraction can be collected. This is also nowadays done routinely in the FEG guns. One has to understand that the nano beam electron diffraction depends on the available intensities of the incident beam, because the sample is particle is not so thin, the diffracted beam intensity may not be sufficient enough to be recorded by the recording device. So, that is why in many cases what is done is that the beams are rather we use the fake beam which is having very high intensity and allowed to fall on the specimen, and then diffracted beam is collected. One of the advantage of this is that one can actually obtain the information for large number of particles by this way, and then get full scale diffraction information from the particles. So, as you see, I am not going to discuss about holography here, it itself is a big subject. As you see that using all these kinds of detectors in the scanning transmission electron microscopy, one can obtain informations encompassing normal wide field, annular dark field, Z contrast images, composition analysis by yields, energy filter images by yields, as well as diffraction information by nano beam electron diffraction. So, this actually seems to like that it gives all kinds of informations, and advantages are many. Obviously, advantages are many, but there are disadvantages. The disadvantage is that the images in STEM mode is obtained in serial manner. So, therefore, it has practical problems like recording the images sometimes can be long, and that is why in that time, long recording time, sometimes images can shift, there will be drift of the images. Not only that, it is also possible that during this taking the images, even the current of the beam, electron beam which is falling from the STEM detector, from the objective lens can degrade as a function of time. So, this can actually give sticky images many times in STEM. Those of you who have used the STEM will find if your microscope is not functioning at the optimum level, you will get sticky images in the computer skin. And not only that, if you use a very highly convergent, high current electron beams, this can damage the sample. Many times you will find if you do nanometer electron diffraction on a particular particle, after taking the diffraction pattern, particle is basically changed. So, the sample damage is very high, that is why in the STEM mode. So, one has to be very careful about the particular specimen, sensitivity of the specimen towards the electron beam. If the specimen is very sensitive to electron beam, one should not use STEM in a FEG gun, rather one should use FEG in the lab 6 filament, where the intensity of electron beam is normally low. So, basically the uses of the STEM depends on that particular type of material. If the material is good and stable, you can go ahead with the normal FEG microscope, STEM configuration and obtain all kind of information. If material is beam sensitive, you cannot do it. So, you have to change to the normal microscope. So, in a nutshell, I have discussed with you the high resolution electron microscope STEM in the last 3-4 lectures. In the next lecture, I am going to show you two important things. One is the in situ electron microscopy, briefly I will discuss, and to some extent I will discuss about the EDS detector, which is normally attached to the microscope. Nowadays, in fact, new concept has come where super EDS is used. Instead of using one EDS detector, one can use several EDS detectors in the electron microscope, which is there in Titan. So, I am going to discuss about that, and then I will move on to the scanning electron microscope, where different kinds of scanning electron microscopic techniques like the EBSD, electron beam, backscatter electron diffraction patterns, or in situ SEM can be used to obtain lots of informations.